Now we are continuing with blueness of stem cells. I will talk about normal stem cells. But first of all, to say something about the physiology and about physiological oxygenation of the tissue, which is, as you know, relatively low. And in general, this is between 3 and 5% of oxygen, and somewhere between 1 and 11% of oxygen. The stem cells are situated in the areas of very low oxygen concentration. In some estimation say that there are also anoxic areas in which we found the stem cells. And if this situation, exposition of the cells, especially stem cells, to the atmospheric oxygen concentration, is the most awful thing to do to the cell, as Professor Brandt said 2 hours ago. But it is still considered as normoxia and as a standard for performing the cultures, which could not be any physiological standard. At the post-capillary level, we found something between 12 and 25 micromole of dissolved oxygen. And this 25 micromole of dissolved oxygen for the cells in general is what somebody call Pasteur point, the point when the cells switch from the oxidative phosphorylation to the glycolysis, to the, from aerobiosis to anaerobiosis. And this concentration of oxygen is achieved with the low oxygen concentration in the tissues, which I'm sorry, I'm using PC in my institution. This one. And this concentration is sufficient to enable the flow of electrons through the respiratory chain in parallel with the transport of the intermembrane space of the protons, which are performing a gradient actioning this turbo production of energy, which is ATP synthase. If there is no oxygen, which is necessary here to cap these electrons and to form water, these two complex, 2 and 3 and 4, are out of game. In that case, we have a possibility to accept the electron with other molecules, mostly by fumarate in the mammal cells, and this reaction going in reverse order is also able to put out the protons and to form some gradient to enable, at some extent, production of ATP. It is very important that the general oxygenation of the tissue was considered by a French scientist, Jean-Charles Massabois, between 3 and 5% oxygen, which is common for all terrestrial and water animals. This concentration of oxygen is established as a standard approximately 600 million years ago in Paleozoic era. And if it is true, and we know today that 2 billion years ago, when the eukaryotes appeared and diversified, the oxygen concentration was approximately 0. And if this standard is concerning the whole tissue, then the stem cell areas in the tissues are corresponding to Proterozoic standard. And this is our hypothesis to compare behavior of the stem cells, of the adult stem cells with the first eukaryotes which appeared on the planet Earth. And we wrote also a book about this, but this is not the purpose today. We put the mesenchymal stromal cells derived from the bone marrow of adult humans in total anoxia in comparison to the culture performed at 20% oxygen. And we found that these cells are proliferating actively and they are expanding their own population of mesenchymal stromal cells. The cultures here presented is until 15 days. If this experiment is performed in presence of glucose at 0% oxygen, the expansion was evident after 5 days, about 3-fold. But if there is without glucose, the expansion was practically absent. Here we have also colony-forming elements between these mesenchymal stromal cells called CFUF, in which population we find mesenchymal stem cells, and we found that they are behaving similarly like the total mesenchymal cell population. When we try to detect and to confirm the proliferation of the cells on the basis of diminution of membrane marker DID, we confirmed that in situation of total anoxia with glucose, we have an intensive proliferation, and we don't have an intensive proliferation. We don't have proliferation at all without glucose in anoxia. But what is very important to say is that in all of these conditions and all of these times, there is no apoptosis, there is no necrosis. The viability of the cells is total. When we performed an assay in which the cells were cultured in anoxia without glucose, that we called here ischemia, because this was ex vivo modeling of ischemia, for 4 days in comparison to the oxygen, to the 20% oxygen cultures, and then we transferred all the cultures in 20% oxygen to enable maximal expansion of these cells. We found that proliferative capacity of the cells previously cultured in anoxia and ischemia was incomparably better and higher than of cells cultured in air. That means that we preserved some of subpopulation of these cells which are responsible for proliferation and expansion. I'm very sorry. C'est ça que je dois toucher. Aha. Okay. Then we try to investigate what's going on with the particular stem cells in these cell populations, and then we perform a single cell assay in order to evaluate the mesenchymal stem cell proliferative capacity and differentiation potential. The cells that were previously cultured for 4 days in 0% oxygen without or with glucose or in atmospheric oxygen concentration were seeded in single cell cultures. And then after growing, some of these cells gave the colonies, the primary colonies. These primary colonies were reseeded in the secondary cultures, and some of them formed secondary colonies. On the basis of this very difficult assay to perform, we could analyze what's going on, which was the capacity of this single cell which produced everything here. And in this situation, we found that the cells from ischemia generated lower number of the primary colonies, but these the proportion of the primary colonies which were confluent was much higher if the cells were issued from an ischemia. That means that the cells issue from ischemia, here for primary, at the level of primary colonies, we eliminated some progenitors while other with a very high proliferative capacity were maintained. In the secondary cultures, the situation was similar. And again, we have a higher proportion of the colonies, confluent colonies, if they were generated from the single cells from ischemia. Combining these results, like the results of differentiation, when a single cell was seeded in a well, and then the colonies which appeared were split in three other wells, and then challenged to differentiation into adipocytes, chondrocytes, and osteoblasts, we can see that the cells from ischemia were all completely trilineage, apart one small fraction of the cells which are about 5% in every condition, which didn't differentiate at all. This is a classical fraction of the cells which were inactivated by manipulation in any experiment in ex vivo, in any approach in ex vivo experimentation. The cells which were in 20% oxygen have, however, important fraction of bilineage and monolineage cells, while the cells issued from zero, from anoxia, exhibited only bilineage subfraction of the cells. This result showed that from ischemia that ischemia maintains only trilineage, thus multipotent, high proliferative potential cells, and this is in line with preservation of full stem cell capacity in this situation, while in high oxygen concentration there is a commitment and differentiation in parallel with maintenance of some subpopulations. But how a cell could produce energy in ischemia? When we performed research on mitochondrial potential, which is reflecting a mitochondrial activity, we find that it was much higher in ischemia than in anoxia, because in anoxia cells were obviously oriented towards glycolysis. But there is no glycolysis here, and the glycolysis was, if there is some glycolysis, minimal. To confirm this, we blocked ATP synthase in ischemia, and we get a diminution in ATP production, which is very important, and it was realized after very short blockage by oligomycin. We tried to block also the complex 2 that we assumed can contribute to the anaerobic mitochondrial respiration, which is most evolution in most ancient way of mitochondrial respiration. And then we used reductase, some agent blocking fumarate reductase, and then we get also an important reduction. And that was the same with the blockage of the complex 1 with rotenone. This approach, however, is not very conclusive, because to measure ATP, the cells should be removed from the condition of ischemia, anoxia, and so on. And we try to confirm this by performing the cultures of the cells, in which we put at the beginning of the culture the inhibitors, and then we observed expansion or maintenance of the cells with or without inhibitors. And when we blocked by rotenone, by pyrvinium pamoate, which is the most classical blocker of the fumarate, NADH fumarate reductase, we found that most sensitive were the cells in ischemia to blockage of complex 2 and of complex 1, which confirmed that there is some important anaerobic mitochondrial respiration passing by these complexes, and probably there is a so-called fumarate respiration in which fumarate is the acceptor of electrons. On n'a pas touché le rond. This was in the culture, which duration was 7 days. And we could see that blockage of any of these complexes provoke a very important negative impact on the survival of the cells, of the mesenchymal cells in ischemia. Then, and they are very preliminary results, we performed a proteome, and we find that the protein complex subunits of complex 1 were increased, enhanced, all of them practically, that we have also enzyme succinate dehydrogenase, which is enhanced, and which is most interesting, enzyme sulfide quinone oxidoreductase and 3-mercaptopyruvate sulfurtransferase, which is one of the 3 ways of H2S synthesis. On the basis of this, we tested the possibility that this H2S way is also important for this respiration in ischemia. And when we added an inhibitor, amino-oxyacetate, which is not completely specific, but however, we get an important negative impact on the survival of the cells in ischemia, confirming the importance of this way, of metabolic way for survival. We designed a hypothetical schema which could be maybe not very academically justifiable, but I don't know. Here you have everything which is blocked and which had a negative impact, obvious negative impact of the maintenance of the cells in anoxia. And now we came to the critical point, fumarate succinate, reduction of fumarate, and acceptation of electron by this fumarate. But in ischemia, where the cells find the fumarate? We found also in proteomic analysis some data concerning some indications of enhancing of glycogenolysis, which could explain providing some glucoses, some pyruvate, which might be go through the malate shuttle, and in relation with this produce fumarate and then succinate. Of course, we are going in the near future to detect, is there any production of succinate in our conditions, which could also be in favor of fumarate respiration. As I said at the beginning, this story started to test the hypothesis that our stem cells could behave in an ancestral way as behaved the first eukaryotes on the planet more than 2 billion years ago. And then more than 2 billion years ago, the ocean was completely, almost completely anoxic, with some ecological areas with oxygen in the traces. It was very low level of glucoses and high level of H2S, and high level of sulfate, pardon. In this situation, the first eukaryote might survive without dividing and maintain their proliferative potential when some minimal oxygen concentration, picomolar oxygen concentration allowed in practically anoxia, but with sufficiently of the glucoses, they could divide it in identical way, and that was the first normal cell division, eukaryote cell division, which is prototype of self-renewal. This is division without any differentiation. And then adaptation to oxygen induced the protection forms of different adaptation modalities of the cells. And finally, we get the single cell eukaryotes with complex cell life, which is resembling to our system of differentiation and commitment on somatic cells, while this first quiescence and self-renewal is referring more or less to this segment of ancient eukaryote life. I don't have the time to go further, but if somebody wants to know what we are talking about, he could find the book and Liryc, 320 pages. It's a little bit, a lot to talk now. I thank you for your attention. Sorry, just to say that this project was performed by my group at the EFS, that the operational project manager was Maria Vlaški, who is here, and the important technical contribution was given by Veronique Labat, and this is my beautiful group. Thank you.
Do you have any question? Stem cell, metabolism?
More stem cell than metabolism.
I have a question regarding this very drastic situation. So no oxygen, no glucose. Is it something we can find in the bone marrow? Did people measure this very stringent condition?
In the physiological condition, I think that you cannot find something like this, but in ischemia could be found in many pathophysiological, physiopathological situations. And I didn't perform the comments, but this work is not only fundamental and conceptual one, it is also a work performing, tracing the new approaches to the conditioning of the stem cells to the transplantation in ischemic tissue. And it is of paramount interest to understand how it works and then to condition these cells by pharmacological approach, by inhibiting, by stimulating, by modulating the metabolism of these cells in order to get good survival. Because today, the most important problem in cell therapy is the mortality of the cells injected in the ischemic tissue.
Is it because of the reperfusion phase?
This is because of exposition, in my opinion, because of exposition of the cells to this extra high oxygen concentration of atmospheric oxygen, of hyperoxia. And then the cells, if they are exposed, they are shocked. If they are exposed long time, they start to adapt. Then you put them in anoxia or even in ischemia, and they are completely lost.
So maybe you should try the sequel and the cycle ROS inhibitors in your cells.
There are a lot of possibilities and ideas, but this is a story which is at the beginning.
Some question? No more questions?
It's too late.
Thank you.
Thank you.